Thank you very much. Good morning, everyone. It's a great pleasure to be here today to share with you part of our research. I would like to thank organizers for giving me this opportunity. I will share with you what we have done in our lab on one single protein, but before that, I would like to share with you a kind of overview of our team. Before I start, I would like to thank all the people who contributed to the specific work that I'm going to share with you today. Richard Gilliland at Synchrotron here at Cornell, Ying Li, Chico Lee, these are PhD students, as well as some postdocs on the right side, as you can see here. So if you haven't visited our department, this is our department in the Tower Road. You can see this milk bottle here. We have the best ice cream in the nation, and I'm not biased, believe me. And this is our team. So everything I share with you today is not my work, but the work of talented PhDs, postdocs, undergraduates in our team. We are working at the interface of chemistry, physics, and biology, soft materials. Anything is squishy, soft materials. Food is soft materials. Proteins, soft materials. So a couple of slides about our team's background, and then I delve into one single protein. What we do a lot of work on encapsulation of actives, whether these are actives that you want to deliver to human GI tract or in chemical processes or even food matrices. So what we do, we encapsulate actives through reversible or irreversible method. We design them to be triggered by external stimuli such as enzyme or pH, as well as external shear stresses. We design also porous materials, anything made out of polymers, or fibers that you can get out of waste streams from, for instance, winemaking processes, pomaces. When we make these type of fibers or porous materials, we actually use them for capturing precious materials from waste streams. Examples here, these are porous materials, woven, cuff, covalent organic frameworks that we make in the lab, but examples are heparin. In meat industry, people use a lot of meat and then we get the mucosa from pig, and we can actually recover heparin sodium as a blood anticoagulant using this approach. We can remove pollutants such as PFAS and other pesticides from water streams. This is an example where we have actually labeled oil and water with blue, which is a kind of a representative of pollutant, and we set the filter, the porous material at the center, and as you can see, we can separate water from the oil and purify water so we can get actually pure form of water without contamination. Same thing with, we've worked with Navy to remove emulsions from bilge water. One other example I wanted to share with you that we are working on sustainability as well as conversion of waste to value. Probably you can recognize this box here. These are termites, and if you look at these termites closely, they are learned and trained to use any sort of cellulose-based materials. And we grab some of the bacteria from termite, we transfect the plasmid, and what it does, this bacteria now can feed on sugars or any sort of waste material such as horse manure, and inside its belly start to produce bioplastics. So we can create bioplastics such as polyesters that can be used for non-contact packaging in the industry. Now this is an example that we use. We work with one of the farms close by to get these type of material, horse manure, and we actually detoxify it, fed it to bacteria, created these type of plastics. You can actually make, even at bench scale, you can make grams or even half a kilogram of plastic, and now you can use this for a kind of non-contact packaging. These are biodegradable, so you can actually use this, and you expect basically having degradation in order of 1 to 5 years. We work also on soft materials, but active soft materials such as cell differentiations. One example is these villi-shaped structures that you can see here, where we actually design human intestinal shape structures that we have villi-shaped structure inside human intestine. The organoids of cells start to differentiate, and you can use this as a model to evaluate nutrients and delivery into gut. We look at microbiome within these encapsulated bacteria and fibers, and we also look at sperm migration, cell migration in basically different application. One example is we work with a lot of IVF clinics to do cell separation for IVF IF and ICSI applications. And whenever needed, we design our own platforms to study, for instance, biodegradation of actives. In this case, we design microfluidic chips, it's just 5 centimeter by 5. We can evaluate degradability of polymers within these chips using machine learning, transparency changes, as well as enzyme treatment inside these chips. I will talk about this high pressure SAXS. Well, we use this type of technology or platform to study proteins, and eventually I want to share with you that we design acoustic tweezers. Using lithium niobate, we design sensors or acoustic tweezers where you can launch sound waves. With the sound wave, you can focus on single particles, manipulate particles, rotate or apply torque, and move them around. So these are type of platforms that we are dealing with. Today, I'm going to only focus on this platform here, one single protein. Can you guess what you're looking at? What this is? Any guess? Very good, Cheetos. And in fact, if you don't color Cheetos, if you don't add synthetic orange color, this is what you get, with a bit of exaggeration. This is what you get. If you add orange color, this is what you get, right? Almost majority of products that we have in the market, in supermarket, jams, you look at strawberry, look at blueberry jams, look at M&Ms, the color is a synthetic form of colorant that we need to use, otherwise nothing will be appealing for consumers to buy. And this is an example. If you look at, next time when you look at the label of any sort of food product that you buy, look at the type of colorant that they are using. Usually if you zoom in, you'll see something like red 40, orange 6, caramel, all sorts of these colorants are synthetic. They are made out of petroleum or oil. These are very stable molecules. They don't degrade. The challenge with natural colorant is that when you put them in the market, in the shelf, the fluorescent light, the light in general, bleach the colorant, the color is gone, degrades over a week. And you know the shelf life for jam, for beverages, is order of 12 months, 24 months. So we have to use synthetic colorant. However, there is a push because there are some studies showing that this could cause ADHD in kids, and there is a good push from the consumers. Can we move from synthetic to natural colorant? Here is one example. If you look at spirulina and you extract it simply with just soap, with alkali solution, you can actually get this vibrant colorant here. It's a natural blue color, the only natural blue color that has FDA approval. So if you get this natural blue color, you can add it to frosting, to Gatorade, to beverages, any sort of active packaging material that you want to package and put on the shelf. The structure is very interesting. It comes with trimer structure. It has an alpha beta and it's order of size of basically 44 kilodalton. When it's a neutral pH, when you extract it, it's very vibrant blue color. Guess what if you want to make acidified beverages, a lot of soda, Gatorade. These are all at pH 3. You acidify it, this is what you get. You get bleach color. You get the color fading away. You want to pasteurize it, you heat it, you get aggregation sedimentation. At pH 3, at pH 3, at pH 7, pasteurization, light exposure, all of these results in a fade of the color loss, so you don't have a product. So we were approached by multiple companies. It's a huge challenge in industry. Can we find a solution to solve this problem, to make this blue color stable? And the question is why this happens. If we understand why, then we will be able to find probably some solutions to make it more stable. So here comes Ying Li. She's one of the best PhD students we had. She's now professor back in China. And she started working with Richard at CHESS here at Synchrotron, and started using SEC, SAXS exclusion chromatography, to fractionate the extract that we get from spirulina. Now, when you do fractionation, you can actually use SAXS to look at different fractions and different assembly, different sizes or aggregates that we have in the solution, if they are homogeneous enough. So you can reconstruct those. And what Ying found was at pH 3, we have this assembly, a smaller monomeric sizes, and that results in a higher exposure to light. And that results in a fading color much faster with a 20 times higher kinetics at pH 3. Now, if you look at here, right side, we have all the protein in the range of 5 to 7. Keep in mind, when we talk about food, relevant pH is 3, acidified food all the way to pH 7. So pH 5 to 7, we usually see well-folded protein. They are more stable than the one that are acidified. When we look at pH 3 to 9, 3 and 9, you see partial unfolding. And in some cases, if you have ionic strengths increase in the solution, you have unfolded, completely unfolded protein. Now, if you look at these under UV, you see a fluorescence. And in fact, you can see a fluorescence on pH 5 here, where has a huge emission. And if you look at excitation emission matrices for these, you can see that for pH 5, where we have higher assemblies of these protein together, they self-protect, they come together, they stack up, and these stacks could be hexamer, nonomer, or dodecamer. And these stacks can protect the protein from radicals, from ROS that generated inside the solution, and that results in a higher stability at pH 5. Also, we know that the isoelectric point of the protein is around 5, so if you look at pH 5, these higher assemblies, in fact, we can evaluate this by accelerated shelf studies, which you take a solution, you put under UV light, expose it for 24 hours. That you can extrapolate for 12 months of shelf life when you put these products under the light in the supermarket. And what we learned is that the kinetics for degradation, for color bleaching at pH 3 as well as 7 and 9 is much higher. In fact, the pH 3 is around 20 times higher than pH 5. Now, to understand why this happens, we did time-dependent EPR, electropyromagnetic resonance. When you do that, you look at the generation of radicals in situ. And what we found is that at pH 3 and 5, in both cases, we get a lot of ROS. There are a lot of huge amount of radical generated within these two conditions. However, pH 5, the protein is very stable. pH 3, it's not stable. One reason is the aggregation, as we discussed, or self-assembly of protein at pH 5. But there has to be some other reasons, because we did some back and forth and we did some control experiments. So I'll share with you why. You can look at this molecule here. This is tetrapyrrole. This is responsible for the vibrant blue color that you see here. The tetrapyrrole in a structure, this is the main chromophore, in the structure of protein, the native protein structure, elongated. If the protein starts to unfold or twist, it will result in twisting of this. Now is elongated form, it can become semi-cyclic. It becomes fully cyclic, and in each of these states, it has a consequence in terms of longevity of this chromophore in certain matrix. So what we did, we actually looked at this structure not only with EPR, but also we look at these byproducts that could happen using LC mass to detect what type of adducts or products we can get there. If you look at the structure of the phycocyanin, we do know there are a lot of tyrosine in the structure, both in the alpha unit as well as beta unit. And tyrosine is notorious for forming dityrosine bonds. So it's irreversible bond formation if you have radical in a solution. So if you shine the sample, shine the product, with fluorescent light or UV, depends on the time, you can get tyrosine bond formation that locks in place the structure, and it's irreversible, can't be dissociated anymore. So that's what we found here. And we looked at these samples, again, non-irradiated with SAXS and irradiated form, that we see elongation happens, and it's kind of permanent elongation. You can't dissociate them under non-reducive condition. So what we learned is that if we want to improve the properties, maybe we can lock in place this structure at pH 5, then go back to pH 3. By locking in place, you can use small molecules, I'll show you how, or you can use enzymatic approach to keep the assembly as is. When you change the pH, you don't let the protein dissociate, and then you can actually keep the structure and the color integrity. This is an example. This is the last slide for this piece of the story. This is an example when we wanted to evaluate and validate the structure. You run STS page. This is 6 hours, 12 hours, 6 hours, 12 hours at pH 5, pH 3 when you shine it with UVA. What we see is that the dityrosine cross-linking happening, and that results in the formation of new band or larger bands, both at pH 3 as well as pH 5. And in fact, there is a specific dityrosine fluorescence spectra that you can record at 405, tells you that the dityrosine forms at both pH 3 as well as 5, and the intensity depends on the structure and the concentration. But we do see that both of these pH 5 and 3, they form dityrosine structure formation. Now, what we learned out of this part of the study, I tried to be very terse, just give you the key points here. We know that if we lock the self-assembled structure at pH 5, then shift to the other pHs, we can actually increase the longevity of the protein. Second, if we add radical scavengers and control the pH by 0.5, 0.3 unit, that helps improving the structure of this protein. And this work was reported by macromolecule. Again, Ying and Richard are really the people who did this work, and we received a lot of support from Chess, from Richard and other colleagues here. I want to share with you a second story, relevant same protein. Who knows about HPP? By show of hand, how many people know about HPP process? A couple of people, perfect. So HPP stands for high pressure processing. In fact, when you want to pasteurize food, whenever we want to buy food from supermarket, it has to be pasteurized. You want to buy food that is pasteurized. Most of the time, it's thermally processed. You heat it up, you kill the bugs, and it's safe. There is another method called non-thermal method, one of the non-thermal method called HPP. What do? You put your samples, your products, inside this blue basket here. You put it inside the chamber, you lock it, you add water, and then you pressurize it. Huge hydrostatic pressure, up to 600 megapascal. When you do that, you crush the bugs, the membranes. The membrane become permeable, you kill them. Now it's safe. In the packaging, you sanitize, you put it back in the market. Now, in fact, this unit is our Cornell unit that we have. It's a HPP validation center at Cornell Food Science Department in Geneva, 1 hour from here. What we do is that you can actually take this approach to also look at protein or biomacromolecule and see how we can use this in our advantage. This is practiced at the industrial scale, but also what we can do is we can look at how pressure induces deformation and whether these deformations are reversible or irreversible, and how we can use this method to make the protein stable. So I'll show you a couple of examples here. The challenge here is that before I share with you how we can take advantage of this method is this unit is so large, and we don't have any sensor, any detector, any way of analysis inside this chamber. It's a huge chamber, just filled with water, a lot of bottles there. How can we look into? How can we understand the dynamic pressure response? If you increase the pressure, ramp up to 600 megapascal, go back down to 0. How you can understand what's going on within that time frame. And again, here comes Richard. If you are here at Cornell, you just walk across the street, talk to Richard, and he helps you with these type of high pressure cell that we have. In the high pressure cell, you can load your sample, and now you can ramp up pH to 350 megapascal. And you can have UV in situ, you can have SAXS in situ, you can evaluate, analyze your sample within this system. So what we did, here what you can see, in fact, on the left side, we ramp up at pH 7, we ramp up the pressure all the way to 350 megapascal, and as you can see, we get alpha-beta monomer. When we start from trimer here, we get to alpha-beta monomer. Okay, that's fine. We go back and we do the same thing. We see the same thing, it's reversible. We do exact same thing at pH 9. We get single units. alpha or beta units. Now, if we do this process, ramping up to 350, going back to 0, we do see that the radius of gyration decreases, the particle size decreases, which is intuitive, we expect that, but also we see some shift, some red or blue shift. In this case, red shift. The red shift that we see in the UV-vis spectra in that chamber when we do increase the pressure up to 350 megapascal is that the chromophore, you remember I showed you that tetrapyral chromophore that is responsible for the color in this protein? That chromophore gets twisted. It's ruffled. It's not in a native form anymore. That twisting or ruffling or changing from the elongated to the cyclic or semi-cyclic, that results in a weakening the conjugation system, more susceptible to ROS, degradation, color bleaching, failure of product. You don't have a product. We don't have a product. How can we do that? Again, based on what we learn, we can find some solutions to address this problem. This chromophore, as you can see, gets twisted, and this red shift here from 0 all the way to 350 megapascal shows that the chromophore becomes relaxed and essentially start to rotate, and that results in a formation of a less strong chromophore in this setting. And AQ or A story essentially is Q band over Sorey band that you can divide, and that number tells you the structure of the chromophore. So if you look on the left side, what we have an elongated chromophore. This is the, again, ratio of Q band over Sorey band. And on the right side, we have this cyclic chromophore that folded and essentially is less stable to any sort of external stimulus. Folding the folding of the tetrapyral essentially is one of the key challenges for the stability of this protein. Now, I told you about the pressure effect. We also learned that we can use pH in our advantage. So we have pressure, we have pH, two knobs, separately or together, we can play with to create the protein structure that we want and make it stable. And so in a way that when it's put on the shelf under fluorescent light, it can basically retains its stability and longevity for 12 months. So what we have done, we came up with a wide range of green approaches, and this is a summary of more than, I would say, 10 papers that I will share with you, the list, but 10 papers that we found different ways based on the understanding that I showed here, we could solve the problem and provide solutions to industry. So it's anywhere from very fundamental understanding what is going on at the molecular structure all the way to the actual application when you take that protein, put it in a beverage, you mimic something like Gatorade, you heat it up, you pressurize it, you fine-tune the temperature and the pH, and then you look at the shelf stability over accelerated conditions. So here comes Jason. Jason Zhang is one of our postdocs, a former postdoc, and based on this information, he started looking at combination of this protein with larger or smaller biomolecules. You remember when you pressure up to 350 megapascal, you get these subunits separated. Imagine if you add a tertiary ingredient or secondary type of protein that results in a binding of these assemblies, bindings of these monomers, when you reduce the pressure, when you release the pressure, and this binding becomes irreversible. If you do that, you can lock in place those monomers, keep everything assembled together, and at lower pressure, you have an ingredient now that you can put inside a beverage that is stable over a long period of time. So he has done that, and he looked at even wider range of pressure, up to 600 megapascal. And what we learn is that if you use whey protein, protein that you find in milk, such as alpha-lactoalbumin or beta-lactoglobulin or BSA, any of these proteins, as well as carrageenan, one of the gums that you see in a lot of different products, you can actually improve the stability. These are very diluted, exposed to light, and we just wanted to show the contrast here. But the whole key point here, we can make this stable, and we can actually create this product. Now some of these are going to be on the market. So we can improve light stability of phycocyanin by complexing with whey protein as well as carrageenan. So this is the last slide, I believe. This is Chico, as you can see. He's very passionate. He's one of our PhD students in the team. And he said, why don't we use this protein not only as a colorant, but also as an emulsifier? Majority of products that you look at, beverages, salad dressings, creams, mayo, they all have emulsifier. Baking products, they all have emulsifiers to make that foam or emulsion stable. This is a protein. Why don't we use this protein to act as a natural emulsifier and also colorant? Now you can make frosting or ice cream or anything that you want to color blue with this protein. So he started this work last year, and he has gotten a lot of cool results. In fact, you can control the interfacial tension at the interface of these droplets using this protein, and you can create not only foams or emulsion that are stabilized by this protein, but also the color becomes more stable because of the understanding that we got from SAXS study. If we assemble them at the interface of oil and water and lock them in place, they are much more stable toward ROS. The chromophore is not exposed to ROS, so it remains very stable. Even with EPR, you see a lot of signals and you see a lot of radicals inside that solution. So if I want to summarize, I hope that I conveyed in a very brief message that we have studied mechanism of degradation. We understand what happens to this one single protein that is the only natural blue currant in the market, approved USDA in US. We found ways to make it stable using HPP, pH, ionic salts, secondary or tertiary ingredients, enzymatic approach to cross-link in place. So it's a wide range of different approaches that we have used. And now we are looking at the surface activity to not only use it as a colorant, but also use as colorant as well as emulsifier, foaming agent, gelling agent, and stabilizer. So this is a list of few of the work that we have done only on phycocyanin, only on this protein. And with that, I would like to take any questions you might have.